Hello, everybody, and wish you all good health and happiness in this new year. I am Vijeta Raghuram, an associate editor of iwonder. iwonder is a science magazine for middle and high school teachers. It features writings about the many dimensions of teaching and learning of science in class and outside it. This magazine is published in English, Hindi, and Kannada twice a year. If you are a practicing science teacher, we invite you to write for us. To do this, please send your idea in less than 100 words to iwonder@apu.edu.in. If you would like to receive updates of each webinar and a free print version of each issue, subscribe to us by following the link displayed in the description of this video. You can also catch us on Facebook. On that note, I welcome you all to our monthly webinars. As you know, on the second Wednesday of every month, like today, we invite authors of an article in iwonder for a live discussion. For this webinar, we have with us Shahid Jameel, author of the article What Do We Know About SARS-CoV-2 from our pandemic issue published in October 2020. Shahid is currently joining us from the UK, where he is a research fellow at the Green Templeton College, University of Oxford. He is a recipient of the Shanti Swarup Bhatnagar Prize in Medical Sciences for his work on human viruses. He has served on various review and advisory committees of the Government of India and the WHO, and is also recognized for his work at the interface of science and society. Shahid, thank you for joining us.
Thank you, Vijeta. Pleasure to be here.
Before we start, let me give our audience a brief overview of the format of the discussion. For the first 30 minutes, I will ask Shahid some questions related to his article. All those who have joined us today are requested to type their questions in the chat box. In the next 30 minutes, we will choose from these questions and questions from a pre-webinar survey that we had carried out. So, Shahid, let's start with the origins of SARS-CoV-2. First of all, why do we investigate the origins of novel viruses like SARS-CoV-2? What insights can such inquiry offer?
Well, we want to know where any pathogen came from so that we can try and limit its introduction in the human population. We also want to know because if a virus came from one animal source, let's say, then there are probably other viruses also that are capable of infecting humans that are present in that animal population. So it's good to know, what, where something that makes us sick comes from a practical point of view. But of course, from an academic and intellectual point of view, it's very important to understand how viruses evolve, how viruses develop in animal populations and then jump to humans. What are the causes for this jump? And therefore we can mitigate them. If we don't understand it, we will not be able to limit it, to mitigate it, to control it.
Okay. Now, in your article, you shared that the scientific consensus seemed tilted in favor of SARS-CoV-2 being a product of natural evolution.
Right.
Maithili K, a teacher from Azim Premji Foundation, asks, some genomic studies indicate that the virus could have been developed in the lab as a synthetic virus and left into the wild. How do we validate that SARS-CoV-2 is a natural virus with zoonotic lineage?
Well, the current scientific consensus is that the virus most likely came from an animal source. Now, with time, this information may change. And that's the first thing to understand is that is the nature of science. Science is not based on absolute beliefs. Science is based on facts and evidence. So the evidence available at this time is that the virus most likely jumped from an animal species, and the animal species is most likely a bat. The reason for this is viruses very close in sequence to the SARS-CoV-2 virus were found in 2018 in eastern China. And these viruses have not really been found in the human population at all. So the, the consensus really is that at some point they infected humans and also evolved in the human population, because there are certain signatures in the virus that are not present in the virus isolated from the bat. Now, I know that people who have this hypothesis that they made this virus in China, uh, the basic flaw with that hypothesis is that, as a virologist, if you asked me to make a virus that is going to infect the human population very badly, my starting point is not going to be a virus which was never known to infect humans. I would start with a virus that infected humans. So I was much likely to tinker with the SARS-1 virus and produce a virus which transmitted more efficiently than tinker with an unknown bat virus never uh, having infected humans. Secondly, when the process of evolution takes place, it takes place slowly. When you do something in the lab, you introduce something, uh, at a go, and therefore it's like a continuous flow versus a disruption. And we don't really see that disruption in the sequence. What we see is that over a period of time, this virus has possibly developed in a human. But of course, as I say, all that depends on whether we are able to find the earliest infected human. And we haven't really found the earliest infected human. All we have is samples from November 2019. Now, if the same virus was found in somebody, let's say in September 2019 or in June 2019, and that sequence is a little different from what came in November and December, that will tell us more about how this virus has evolved. So it all depends on that. And finally, I think this debate, whether it was a man-made virus or whether it jumped from animals, will continue uh, unless, for example, China, which is accused of doing this, opens all its computer records, all its hospital records, all its laboratory freezers for public inspection. We all know that that's not going to happen. So this debate will rage on, and my prediction is 10 years from now, 20 years from now, we still we'll still be debating this. Uh, I think the more important thing to do is focus on controlling this rather than worrying about where it came from or where it was made. But I think it's very, very important to put some limits on how viruses can be tinkered in labs. Uh, because if you tinker viruses in lab and you make the virus more infectious or more pathogenic, and that virus somehow leaks because your lab didn't have the adequate contamination protocols, containment protocols, then that could be a problem. So I think the world needs to look at this seriously. This whole issue of uh, whether people can make a virus in a lab. And by the way, I mean, I'm giving a very long-winded answer, but I also want to emphasize that making a virus in a lab is not something that is very difficult. You know, A virus was chemically synthesized almost 20 years back. Small pieces of nucleic acid were purchased from commercial suppliers, and a whole polio virus was synthesized from those chemical components. This happened in an academic university lab in New York State in US. So the technology is there. It's how we regulate this technology ourselves. That's very, very important.
Okay, so to sum it up, while it is possible to create a virus in the lab, and there can be a whole discussion about the regulations around it. As far as SARS-CoV-2 is concerned, we do not have any clear evidence that it was generated in the lab, and all the evidence so far points to a natural evolution.
Absolutely.
Today, SARS-CoV-2 is in the news mainly for its variants.
Mhm.
So, speaking of evolution, how do variants arise?
Okay, variants arise because uh, of mutations. Mutation is a change. So a virus has a nucleic acid. Nucleic acid, that's the genetic material of the virus. In this virus, it's the RNA. Now, when a virus infects our cell, it makes more viruses. How does it make more virus? The genetic material multiplies. So if one RNA is there, it makes two molecules of RNA. Two makes four, makes eight, and that's how it multiplies. Now, this multiplication is done by an enzyme which is called a polymerase. Now, this enzyme, while it is reading the RNA chain to make a duplicate chain, sometimes makes errors. And these errors cannot be corrected because this enzyme does not have any proofreading activity. It cannot go back and change, it only moves in one direction. So, when a change happens, that change gets embedded in that genome. So, let's say it started with a template that is, let's say, all blue. It made another strand which is all blue except one position becomes red. Now that red becomes embedded in that chain, and that, when that chain with the red makes another chain, maybe at another position it makes a chain that is green now. So what you get is all blue plus one red plus one green, and that's how mutations propagate. Now all mutations are not useful for the virus, and therefore we don't see them at all. But some of these random mutations become embedded in the genome because they offer some advantage to the virus. And when multiple mutations come together in a given lineage, that becomes a variant. So the variant virus is different at several positions in its genome from the original virus. That's how variants arise.
Okay, so now Vinay Suram, a teacher educator from the foundation, asks something very interesting. How do we identify a new variant, and how do we conclude that it is a new variant and not a new virus? Like you said, variant must be having a lot of new changes to it, right?
Right, right.
How do we conclude?
Okay. Well, think of it this way, we are all humans. We have a genome that is indicative of us as humans, but all of us don't look alike. So we are sort of variants of each other. But, a human genome cannot produce a bird. That's a that's a different species, right? So think of it that way. The way you find out whether it's a different variant is by completely sequencing the genome of the virus. And when you sequence the genome, you find that it is different at multiple positions. And virologists have a way of doing it, because if a virus, if one virus differs from another by a certain percentage, you call it a new variant. uh, but the working definition of a variant really is if it causes a disease that is slightly different from what the original virus was causing. If it has some phenotypic properties that are a bit different, maybe it's transmitting a little differently, maybe it's evading, neutralizing antibodies a little differently, then it becomes a different variant. But the way you de- decide whether it's a new variant is based on sequencing, and sequencing only.
Okay, so there seems to be a range of differences, right? If
Yes,
so
yes,
much difference happens, then it's a variant.
yes.
But beyond a certain level of changes, it's then to be considered as a whole
Well, I
different.
mean, SARS-CoV-1 virus and SARS-CoV-2 virus are different viruses. They both belong to the same family of viruses, but they're different viruses because the genetic identity between these viruses is only about 80%. The genetic identity between two variants of uh, SARS-CoV-2 is very high. It's, beyond 98, 99%.
Okay. Now, the WHO and CDC classify SARS-CoV-2 variants as variants of interest, variants of concern, and variants of high consequence. What criteria do we use to make this classification? And can we predict what kind of mutation is likely to be of high consequence?
Well, the way WHO does this, if they so firstly, there is genomic surveillance going on across the world. Regional and national governments, private academic labs, many people are sequencing viruses, and they are depositing these sequences in public databases. So by now, about 7 million sequences have already been deposited in public databases. This is continuously being monitored. So, for example, I'll tell you how Omicron came about. In Omicron was first seen in 4 samples from Botswana, and then in South Africa they found the same sequences coming up. So they reported this to WHO and said, well, this is spreading very quickly, and we feel that this should be called a variant of concern. Now, although it was the call of WHO has normally been calling something a variant of interest if they find that a new variant is spreading. But since this variant showed almost 50 different mutations compared to the original virus, and it was spreading very quickly, and these mutations were getting mapped to a place where people thought that they would evade antibody responses very significantly, the recommendation of South Africa was call it a variant of concern right away. And the WHO committee met and decided, yes, they're right, we should do that, and they called it that. Normally, they'll label something as a variant of interest, and if they find that it continues to spread, if it continues to evade, they will upgrade it to a variant of interest. That's what a variant of concern. That's what happened to alpha, beta, gamma, delta. But Omicron is a little different in that sense. Yeah.
So, before we move on to Omicron, could you give us a brief overview of the SARS-CoV-2 variants that we know of now?
Yeah. So there are actually many variants. So and this list of variants of interest and variants of concern is a shifting list. It's not really a list that's in perpetuity. So, for example, many variants have been listed under variants of interest, but then they have been dropped from that because they didn't move up to the variant of concern. So at this point, there are 5 viral variants that are called variants of concern. It started with Alpha, which was originally the variant discovered in UK, or also called the Kent variant earlier. Then came Beta. Beta was the one that was found in South Africa earlier. Then Gamma was sorry, Gamma was first found in Brazil. Then Delta, which was first seen in Maharashtra in India, and Delta is the one that caused the second wave in India, and Delta is still spreading around the world. And then comes Omicron. Now, all of these viruses have unique mutations. There are mutations that are present in some but not the other. And you know, in Delta as well as in Alpha, there are mutations that uh, cause the virus to multiply faster and to infect better. In Beta and Gamma, those mutations are missing. So Beta and Gamma haven't really spread that much. However, Beta and Gamma have mutations that are able to evade antibodies very well, which means that if you've had a prior infection and developed antibodies, or if you've been vaccinated and developed antibodies, uh, the virus can evade that. Delta has both virus, immune evasion mutations, as well as a mutation that makes it spread very quickly. And that's why you see that Delta has completely taken over the world ever since it originated. It displaced all the other variants. And now comes Omega, Omicron,
Omicron.
which is spreading even faster uh, than Delta spread, because it has more mutations that can allow it to multiply and transmit better. But the thing with Omicron is that it has brought together many of the immune evasion mutations individually found in the other variants into this one variant. So Omicron now has possibly all the mutations that we have seen in all the other variants. So it's like a super variant, if I may call it. It's spreading very fast. The antibodies are not controlling it as well, but that doesn't mean it will cause more severe disease, because disease is something very different. Disease is something that is how our body reacts to the virus. And the other thing that we have to realize is that when a virus accumulates so many mutations, it also causes a fitness problem for the virus. Uh, and that is what is happening to Omicron. Omicron is not able to cause as much disease. It doesn't really uh, activate the immune system to a level where bad disease manifests itself. So it's a mixed bag right now. Omicron is transmitting very well, spreading very well, but in general, it is causing milder infection. So that's like a 2 minute summary of the variants.
Okay. I'll come to that point about Omicrons again shortly. But first, how does a positive patient find out if they are infected with Omicron or some other variant?
That can only be done by genetic sequencing. The PCR test will not tell you that. Now, there are some PCR tests which fail to recognize the surface gene of Omicron, but they detect two other genes present in the test. And you may have seen something called the S gene target failure PCR. So that's like a surrogate test for it. But if you want to ensure that you have Omicron, genetic sequencing is the only way. And you can't just order genetic sequencing. Uh, this, of all the samples that come PCR positive, public health authorities decide how many of those samples will be sequenced. India is typically sequencing about 0.2, 0.3% of all the PCR positive cases. But for an individual, it's really academic whether you have Omicron or another variant. You shouldn't worry about that. What you should worry about is that your disease remains under control. That's all that matters.
Okay. Now we have data from Omicron in the UK and US, where it is becoming the predominant variant,
Yeah.
and Omicron in South Africa also. Now, what predictions can we make for India?
Well, India is already, uh, India's third wave, the Omicron wave, has already started.
Okay.
The predictions that I have seen coming from two different models, I'll tell you. Uh, there is a Sutra model which IIT Kanpur developed, which the government of India follows quite closely. And the Sutra model in early December had predicted an Omicron wave in the worst case scenario to pull about 150,000 cases per day at peak by the end of January. Now, we know that India is already pulling somewhere around 160 to 180,000 cases per day, and we are still, around a little before the middle of January. The other model, which is the Indian Institute of Science, Indian Statistical Institute model, which was released on the 10th, 2 days back, presents three different scenarios. In the best case scenario, they are showing about 300,000 cases per day at peak, and that peak will come by the end of January. In the worst case scenario, they are predicting somewhere around 800 to 900,000 cases per day at peak towards the middle of February. Uh, so at this point, it looks like the ISI model is probably on track. Uh, we just hope that it's their best case scenario prevails. Uh, India will get a lot of infections, uh, many more infections than we got in Delta. Uh, but the situation is very different now because much of India has already been infected in the Delta wave, and many of them have also received at least one, possibly two doses of vaccine. And therefore, the disease is going to be mild. We are not going to face the mayhem that we faced with Delta. So don't be scared, just live it out.
Okay, I'll come to the point of the vaccines shortly, but there was something else that caught my attention. You mentioned that the third wave, the Omicron wave, is now in India. So, is Omicron alone driving this third wave? And do we no longer have to worry about other variants like
No,
Delta?
we can't say whether it's Omicron alone.
Okay,
Sorry, I should not have used the term Omicron wave. I should have just said the third wave.
okay.
Because even now, you will be getting infections of both Delta and Omicron. Those are the only two variants present in India. Uh, it's likely that since Omicron infects much better than Delta, and there are estimates that it infects, 20 times, 30 times better. But so based on that, most of the infections are likely to be Omicron infections. And that's what sequencing is also showing, that most of the infections are Omicron. But Delta infections are still there, absolutely.
Okay. And how do we live this out? I mean, could you briefly outline some key measures that governments, public health systems, educational systems, and individuals can take to minimize the impact of the third wave?
Well, action at multiple levels. At the individual level, what you and I should do is, firstly, wear a mask and wear it properly. Uh, Stay more in ventilated spaces than in closed indoor spaces. Uh, Even if you are at home, it helps to open a window. If you're in an office, open a window. At least these days, you're not running air conditioners, so that's good. uh, Also, avoid going to places that are very crowded, like markets and all. If you can avoid it, avoid it. Uh, any indoor activities which require you to take off your mask should be dealt with extra caution. So if you are in a closed space in a restaurant, you have to remove your mask. I mean, you can't eat with a mask. So that I would consider to be an at-risk activity at this point. So just use common sense. Uh, as far as the public health response is concerned, I think the time is gone for uh, trying to contain the infection. We can't contain the infection. So, what we have to do is to ensure that hospital capacity is available for those who need it. Most people will not need to go to a hospital, but capacity should be there for those who have to be there. Um, and of course, vaccination has to be speeded up, and people who are in the vulnerable age group, people who are elderly, have to be given booster shots on priority. So those are some of the measures that have to be taken by individuals and public health response.
Um, Now, let's, there are some questions related to vaccines that are coming up. Um, First, Vinay asks, why do symptoms of, I mean, I think in order to understand this need for a certain age group to get vaccinated, um, why do symptoms of COVID-19 vary so much? And why are a few people asymptomatic while others die from COVID-19?
Well, humans are different. Humans are not inbred mice that we use in labs. Each of us respond differently. Some of us have to take 2 pills of paracetamol for our headache to go away. Others don't need any. Uh, so we differ in our response to everything, and Omicron, COVID is no different. Uh, Symptoms are based on how we react to the virus. The virus is the same, but how our immune system, our metabolic system reacts to the virus is different. So some of us are more prone to upper respiratory infection, and we can limit the infection there. In others, the infection goes into the lungs, and that's where it causes most of the problem. So the virus is not different. It's us who are different, who are responding differently to the virus.
We have a question from Jason Fernandez, who asks, "Is the virus life-threatening for young people with COPD and CKD and who are on immunosuppressive medication?"
Yes, uh, the disease can be quite severe in people who have COPD. COPD is chronic obstructive pulmonary disease, so the lung is already weaker. CKD is chronic kidney disease, and people who are on dialysis, for example. So yes, these are conditions where people are more prone to infection, and people are more prone to severe disease. Immunosuppressive medication, especially people who are on cancer therapy, are on immunosuppressive medication. People who have HIV infection, for example. So the immune system doesn't raise the right kind of immune responses to control the virus because it's it's immunodeficient. Uh, and these people really need to be watched very carefully, and medical help sought if needed.
Now, many of us in India have taken 2 doses of vaccine in order to prime and boost our natural immunity against COVID
Right.
19. Vinay also asks, how long does this protection last and how do scientists measure it?
So it depends on what you mean by protection. If you mean protection from infection, then protection from infection wanes fairly quickly for a variant like Omicron. And studies have shown that within 3 months or so, the antibody levels in our blood go down sufficient that we will not be protected from Omicron infection. But if you mean protection from disease, that's a different ballgame altogether. Because what vaccination also does is not to just raise antibodies, but to raise T cells. And T cells are the ones that kill virus infected cells and prevent further amplification of the virus, which limits disease. So the T cells seem to be working just fine. And in India, 90% people have received Covishield, which is really the best vaccine in class to raise T cells. It may not be the best vaccine to raise antibodies, but it's a very good vaccine to raise T cells. So the disease is going to be, very mild, if at all, in people who have received 2 doses of the vaccine. When you get a booster, what the booster does is boost your T cells, but it also boosts antibodies. So it can provide that temporary relief against infection as well. And therefore, boosters should be given to people who are more vulnerable, like people who are elderly, those who have COPD, CKD, immunosuppressive therapy, all of that.
So basically, the protection against infection can reduce, but protection against disease can perhaps be maintained
More long
through.
lasting, more long lasting. And see, that's the purpose of vaccination. When you vaccinate a healthy person. The person has no infection, no disease. You vaccinate a healthy person to protect from a future encounter with that virus, right? Now, if that future encounter happens a year later, by that time antibodies are all gone, so infection will still happen. There are very few vaccines that will provide protection from infection, but all vaccines provide protection from disease. And that's an important concept to understand. Uh, as long as I am protected from disease, it's fine. I don't worry about getting infected, as long as my immune system kicks in quickly and controls the virus. And that's what happens in an immunized person.
Okay, so is that the reason why we are seeing a steep rise in infections worldwide despite vaccination? Like, uh,
Well, it doesn't have to be just spike vaccination. I mean, the steep rise is because the variant that is circulating is so infectious, and because it is evading pre-existing antibodies. And in India, for example, most people have had their second dose 5 6 months back, so they already have no antibody based protection left, and that's why they keep getting reinfected. This vaccine was not developed to prevent infection. This vaccine was developed to prevent disease. So you often hear people say that you know so and so got both doses but still got infected, the vaccine is not working. That's not the right question to ask. The right question to ask is the disease still mild in that person? Uh, and if the disease is mild, Imagine what would have happened if that person had not taken the vaccine. The disease would have been severe. Uh, this, please understand that this vaccine is not made to prevent infection. It is made to prevent severe disease.
And with the additional dose, again, we are sort of increasing our protection against
Yes,
the
against both
disease.
infection in the short term and disease in the long term.
Okay. Okay. Now, there is one question related to, okay, Chandra Siddh asks, what does being asymptomatic mean in terms of the biology of the infection or disease?
Well, asymptomatic simply means that you're not showing any symptoms. It means that you got infected, uh, and the way you know that you got infected is you got tested and the virus was found. Otherwise, you won't even know. An asymptomatic person can only be called asymptomatic when they are tested randomly for something else to find that the virus is there. But they're not showing any symptoms, so it simply means that the virus infected, but our immune system prevented it from multiplying extensively in our body and causing disease. That's all it means.
Okay. Now, Anita, a higher secondary school teacher from Rishi Valley, asks, could you throw some light on our current understanding about COVID vaccines for teenagers and children? If you could give a
Well,
brief outline.
in India, there are two vaccines that have now been approved for teenagers. Well, not just for 12 to 17. Uh, One is Covaxin, and the other is Zycov-D, which is the DNA vaccine. Both vaccines have been tested in children and they have been approved for children in India. Right now, I believe it's only Covaxin that is being given to children, but I think Zycov-D will also be rolled out soon. Uh, for children, there is no vaccine yet. Uh, so anything beyond below 12 years, there is no vaccine. Uh, Several Western countries have started giving RNA vaccines to children. I think China has started giving some of its vaccines to children, but in India, we don't have anything like that. And really, I mean, the infection in children is so mild that you really don't need to try and prevent infection in them using a vaccine. Uh, so I think the policy right now is fine. Uh, Look at the science behind it. If you look at the disease severity, it sort of follows a U-shaped curve. And the bottom of that U is the age group of 5 to 10 years, which essentially means that children who are 5 to 10 years old show minimum risk of disease. On either side, the curve starts going up. So, which means that children below 5 years can get uh, more disease, and children above 10 years can also get more severe disease. Now, we have a vaccine that will hopefully cover children largely from 12 to 18. So those are the children we need to worry about because they may get more severe disease. There are also children who go to schools. Children who are less than 5 years are not in schools, so they can be protected much better. So I think that's the situation right now. And if your child is eligible for vaccination, please do go and get your child vaccinated. That is very, very important.
Okay. Now, we spoke about the lifetime of a vaccine and how its efficacy in protecting against the infection can decline over time. But how effective are the current vaccines against the currently known variants? When it comes to variants, what happens? And how likely is it that we will need new vaccines as more variants emerge? This is a question from Maithili.
Yeah, so no, I think it's a very good question. The my sense is that we will need new vaccines against the emerging variants, uh, because Omicron is not the last variant you're going to see. There will be more coming, and I think vaccines will have to be upgraded. But the vaccines that are present at this time are actually protecting very well from disease. They may not be protecting very well from infection, but they're protecting very well from disease. Just look at the experience of every country that has vaccinated heavily, and you will see that while case numbers are going up, hospital admissions are not going up. Uh, and even those people who are in hospitals, if you start seeing this, almost three-fourths of the people are those who are unvaccinated. And about a quarter of those are people who have uh, received only one dose of vaccine. Those who have received two doses of vaccine and those who have received vaccine two doses plus booster, you hardly find those people in hospitals. So this is clear evidence that vaccine is working. And this is not the experience of one country. These are multiple countries are showing this. UK is showing this. US is showing this. US is pulling about 800,000 cases per day. But if you look at their hospital situation, hospitals are not overflowing like they were earlier. And this is US data that three-fourths of people in US hospitals are those who refuse to get vaccinated. I mean, what other evidence do you need to show that vaccines are working?
Now, there's a question from the audience. Ganga Rotela asks, there is an opinion that Omicron may serve like a vaccine. How much is this justified?
I think that's a very irresponsible statement that Omicron is a natural vaccine. Uh, because how the disease is going to manifest depends on an individual. Somebody who has comorbidities, somebody who's elderly, will show a more severe infection from Omicron compared to somebody who doesn't. So that statement means that you actually encourage people to get infected with Omicron because that will work as a natural vaccine against any future variants. I think that is something that is very irresponsible to say. We shouldn't be saying this. We also don't know what the effect of long COVID is going to be, even in somebody who is, uh, you know, who has very mild infection. So, based on all that, I think prudence demands that we continue to get vaccinated and protected rather than treating it like chickenpox, where one kid on the block gets chickenpox and every other kid is sent to meet that kid. Uh, don't treat Omicron like that, please.
Shahid, can vaccines contain the evolution of new variants?
Sorry, say that again.
Can vaccines contain the evolution of new variants? If
What vaccines do is limit the multiplication of the virus in an infected person. And if the virus multiplies less, it is going to make less errors in multiplication and therefore limit the variance. So the more the virus multiplies, the more the virus circulates, more are the chances of variance. Uh, so vaccines indirectly do limit the emergence of variants.
so, it's clear that getting vaccinated should be an important priority, like,
Yes,
or
not
rather
just
the.
a priority, you owe it to your health and you owe it to public health. It's it's not just a responsibility, it's also a duty.
Um, there is a question from Asif Iqbal: how to improve vaccine equity to restrict further mutation of virus?
Well, you can't improve vaccines to prevent mutation. Mutation is a random process. When the virus infects and multiplies, it will mutate. You can't do anything about it. It just happens. So, as I said earlier, if you are vaccinated, the virus gets very little chance to run away with uh, multiplication in your body, and therefore you will naturally reduce mutations. But you can't make vaccines to reduce mutations. No.
I think there's
Oh,
one.
sorry. I'm sorry. His question was improve vaccine equity.
Yes, actually, I
Oh,
think it is based on what your
right,
earlier
right. Sorry.
response that.
Yes, so I think it's very important question. Vaccine equity is very important. If parts of the world are not vaccinated, the virus is running wild in those populations and mutating. And look at Omicron, and then there's another variant which is still a variant of interest, is called B1640.2, which was first seen in Cameroon in Africa. Omicron came from Botswana and South Africa. All of these countries, whether it's Cameroon or South Africa or Botswana, have high levels of HIV infection, meaning immunodeficiency. They have low levels of COVID vaccine coverage, and the most likely origin of these heavily mutated variants is that they continue to persist in an immunocompromised person and continue to accumulate mutations. If those people were vaccinated, the virus wouldn't continue to multiply. So, at a population level, vaccine equity is very, very important, extremely important. And I think the world needs to look at it. While parts of the world are already on their fourth shot, Israel, by the way, is giving fourth shot to everyone.
Oh, wow.
Uh, there are parts of Africa that have only 1% vaccination. So we need to look at this very, very seriously.
And is there a discussion going on about how
If
to
there is
increase
discussion,
that?
WHO has been very vocal about it. WHO chief has continuously said that if we are not making vaccines accessible to Africa, more variants will emerge from there. And it does appear to be the case.
Okay. Um, One question from Subramanian VL. He asks, does it mean that, okay, I think this is related to the variants actually. Um, can a person infected by Omicron not be infected by Delta variant?
No, that's not true.
Okay,
You
they
could be
can.
infected by both at the same time. If you mean if you have had an Omicron infection, can you then be infected by Delta? Well, it depends on the duration between Omicron infection and Delta infection. Uh, so, if you have had any infection and it's been 6 months since that infection, your antibody levels are going to go down to a point where a fast moving variant like Omicron will infect, or a fast moving variant like Delta will infect. So that is quite clear by now. But
Okay.
if you are exposed to Omicron and Delta at the same time, you could be infected by both at the same time.
Okay. Now, two terms that are used frequently with SARS-CoV-2 are herd immunity and endemicity. What do these mean?
Okay, so herd immunity means if there is a small percentage of people who are unvaccinated, can they be protected by the larger herd which is vaccinated? So let's say in a room we have, 50 people. Out of those 50 people, two are unvaccinated, 48 are vaccinated, and a virus enters that room. What would be the chance of that virus hitting those two unvaccinated people? The chance is going to be fairly low. But if in that room you have 10 vaccinated people and 40 unvaccinated people, then the chance of a virus hitting an unvaccinated person is going to be high. So it really relates to, what level of protection in the community is needed to protect those who remain unvaccinated. And unfortunately, for such infectious variants and viruses, the herd immunity threshold is very high. So let's give you an example of measles. Measles is an extremely infectious virus. And to protect somebody in the community who's unvaccinated for measles, you require 99% of the community to be protected. Uh, so that level of vaccination is needed. Uh, so in this case, I really think with more infectious variants coming all the time, uh, let's not worry about herd immunity uh, at all. Uh, let's all get vaccinated so that we can continue to protect those who cannot get vaccinated, not for their religious or cultural beliefs, but because of their medical conditions. Uh, I mean, that's very important. The second thing you talked about was endemicity. Endemicity means when a virus reaches a point where it's always present in the population, uh, as opposed to, a virus coming in and causing a lot of infection. So endemic simply means it's always there, right? Uh, so this virus is going towards endemicity. It will eventually become endemic, just like four other coronaviruses that cause the common cold are endemic. Before this virus came, you had never even heard of those viruses. They come, they cause a little cold, and you're done with it. Every 3 4 years you get infected by one of those coronaviruses, they cause a common cold, and you never hear about them again. The same will happen to this virus. But how long it will take for this virus to reach that level is the key question. And I don't think anybody can give you a prediction of that. Uh,
Okay.
Most likely in the next few years, it will become endemic. But I would not like to put my money on whether it's going to be 3 years or 5 years or 10 years.
Okay. As we're running out of time, I think I'll ask one final question, uh, particularly in light of the fact that our magazine is for school science teachers. From what you have seen regarding the response to the pandemic at various levels, what are some of the things that may be useful for students to learn in school about the perspective and practice of science in general?
Well, as I started out by saying, science is a process. Science is not a belief system. Religion is a belief system. Science is based on doubt. Uh, Religion is based on beliefs. Uh, so science is based on doubt. Science is based on questioning. Don't believe what I'm telling you. go back and check it. You should always be questioning. That is what science is. And science is a process. Science depends upon evidence, science depends upon data. And for that, it is very important that public policy is driven by evidence and data and not by what someone believes in. So that is science. And I think students need to uh, understand science not as a collection of facts, but go behind the process of how that fact came about. Uh, how was it discovered? What was the process? The other thing about science is that scientists often tend to be contradicting each other. The same scientist may be saying something now and something else later. It doesn't mean that I was lying either earlier or now. It simply means that I have better data, better evidence to support what I'm saying now. And what I'm saying now may change tomorrow based on more evidence, more data. So science is a continuously changing landscape. And that is an important point to understand. Uh, it's not important that the value of the G force is 980. That's not important. What is important is how you determine it. And in any case, that G force of 980 is not exactly 980 at every location on Earth. It varies. So it's important to understand how that came about. How was it determined? What was the process? So think of science as a process. Don't think of it as a collection of facts. That's what I would tell the teachers, and that's what I would tell the students.
Thank you so much, Shahid, for that message. So with that, I think it's time to wrap up our discussion today. Shahid and all those of you who have joined us today, thank you so much. We may not have been able to take all your questions, so apologies for that. If you have any pressing questions for Shahid, please send them to us at iwonder@apu.edu.in with the subject line question for Shahid. We will do our best to get them answered. Join us again for our next webinar on the 9th of February. And for all those who have not subscribed to iWonder yet, we hope you do so now. Thank you.
Thank you very much, Vijeta. Thank you.
Thank you, Shahid.